In this video we will discuss about this RNA sequencing overview. So what are the different approaches that are available to sequence RNA, and what are the advantages and disadvantages of them, that we will cover here. So how uh, the different RNA approaches work is that first the RNA fragment that is need RNA fragment is been isolated, and then in one approach it is converted into, in some approach it is converted into cDNA, and then it is connected to the adapter, which depends on the kind of method, downstream method, that will be used to analyze the RNA. After that, this amplification of this uh, cDNA occurs, and then about which size or what size of the base pair needs to be read is been detected, whether it is a short read RNAs or long read RNAs, and then sequencing is carried out. And so the black line represents the short read RNA, and the green line here, it represents the long read RNA, where in the short, what happens is that there is a RNA is converted into small fragments, whereas in the long read RNA is not converted into a fragment, and it is uh, whole RNAs with whole RNA's complementary DNA has been synthesized and then formed and then further processed for sequencing. Whereas in RNA, what happens is that it is not been converted into the indirect RNA sequencing. What happens? It is not been converted into the cDNA, whereas on direct adapter ligation happens, process carried out based on the type of downstream process that will be used, and then uh, it will be processed for sequencing. So here we will discuss what are the direct RNA sequencing methods that are available. First one, and the most popular one, is the Illumina method. What happens is that RNA has been RNA, or we can say DNA. It can work on both the things. So here it is shown for the DNA. So what happens is that there is a flow cell that contains some sequence oligonucleotides, and the samples are prepared in according samples are prepared in a way that it will bind to this oligonucleotides on the flow cells, and then the fluorescence labeled nucleotide will bind to the complementary end of the uh, sample, and this fluorescence signals has been recorded. As one of the example is shown here, the one of the uh, signals, that green signals represent A, red signals represent T, blue represents C, blue represents C. So this is how later on it has been deciphered what signals are been, what is the sequence of the RNA that is been processed by this instrument. Uh, Quite similar kind of mechanism is been carried out by Pacific Biosciences. In this case, what happens is that instead of binding the RNA directly onto the flow cells, it makes a it makes a round shaped RNAs, and later on it will pass through the this instrument, where the fluorescent labeled nucleotide has been identified using this instrument. And the example is shown here, that green is represented by A, and then T represented by red, C represented by blue. And what happens in the Oxford Nanophore is that the protein motor protein is bind to the motor protein is bind to the RNA samples, and later on this motor protein will help in help or prevent the translocation of protein on this Oxford nanopore and move the proteins in a way that will leads to generation of the current, and this generation of current is been recognized by Oxford nanopore, and it varies for all the 4 nucleotide, and based on this current signal they can identify which nucleotide is been read at present. These are the some basic overview about different RNA sequencing methods available. So now let us understand how the gene is present. The gene is present in this form. It contains both exon and intron. Intron are the non-coding sequence. And what happens during splicing is that different forms of this gene can be formed. Two examples are shown here. One is transcript X, in which this exon is missed by the splicing enzyme, whereas in transcript Y, this exon is included and this exon is missed. So what happens on the what problems might cause during the short read DNA is that there is an ambiguous sequence that might get from both the transcript, and it is not been able to clearly identify which read belongs to which transcript, and these are the examples of unambiguous exons. As in this case what happens is that exons are the short read DNA belongs to only, and short read DNA belongs to both the short what happens in this case is that short read DNA belongs to only this transcript Y, as it is shown by orange color, as they are modified at this end, and same thing is available at this side and this side. So and one of the reason why these are the ambiguous, the correct reason why these are the unambiguous exon is that they are present only in one transcript, whereas in this case it is difficult to identify which exon belongs to which transcript. And so these are the ambiguous isoforms, and these are the non-ambiguous isoforms that software can detect. However, what happens in the long read is that since the complete cDNA has been analyzed, the whole isoform can be deciphered with more accuracy, and it will not have any ambiguous region in case of long read cDNA. So what things that are missed in this long read cDNA is that the modified section of the RNA and the length of poly A tails is missed, since this cDNA is the complementary version of the RNA that is to be analyzed. So the best way to analyze RNA sequence is the direct RNA sequencing. In this case we will be able to see the methylated form of, or modified, not methylated, the modified form of the RNA, and also can estimate the poly A tails of the particular RNA. So this is some of the basic overview of RNA sequencing and what are the different strategies available and which one is best if one needs to analyze for RNA sequencing. That is, direct RNA sequencing is the best method to analyze the RNA sequencing RNA data. I hope you got some overview about this RNA sequencing. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.